your event this week. So thank you so much for having me. And as Beck mentioned, I am in the PhD program down here in San Diego. And I'm in my 4th year, which I never would have imagined would happen. But I was raised in Bothell, Washington, and grew up there, spending over 30 years in Washington before coming down here for school. And it's a little too dry down here for me, so I'm coming right back up as soon as I finish to the Pacific Northwest. So many of you may have seen the title and thought, Antarctica, there are no trees in Antarctica. How is this related to forests? And that's what I'm hoping to do today, is to introduce you to the invisible forest, as us phytoplankton nerds like to call it, that's living in the ocean if you don't already know about them. And before I get into that, I wanted to kind of explain how I got involved in this work, because nobody, at least I've never heard anybody say from a young age they had always dreamed about studying phytoplankton. So kind of taking you through this circuitous route. Let me share my screen so you can see my slides. Okay. And I hope you are all able to see that. We're going to start back in high school, when everybody's trying to figure out what they want to be in life. And I just thought being an astronaut would be cool, because they travel to the moon, which is the most amount of travel anybody could do. And I really loved traveling at this age. So I looked up what astronauts got degrees in, and a lot of them studied STEM or science fields. So I decided I would pursue biology and geology, because why pick one thing when you can do all of it? I then went to the University of Washington, which mostly drew my attention because of the marching band. You know, at this time in our life, we're just making very simple decisions. So the marching band was really what attracted me to the UW, and it was in-state, which is always a bonus. And it wasn't until my senior year that a professor I had in genetics said, if you want to be a scientist, you need to work in a lab. And I thought, oh, I don't know if I want to be a scientist, but I guess I should get a lab job. So I ended up having an internship and finding work. And the internship was at the Allen Institute for Brain Science. I think they renamed it just the Allen Institute down in South Lake Union now. This was in Fremont at the time. And I cut up little ice cubes of mouse brains into thin sections and scanned them into the computer with fluorescent labels, and then constructed models, or 3D models, of the mouse brain and where different genes were turned on or off. And this was way too much computer work for me. So I was like, this is cool, but I've got to find a way to get outside. And there's actually now online, free to the public, if anybody's interested in brains, Human Brain Atlas and the Mouse Brain Atlas. So I didn't find a job outdoors. Instead, I ended up working in a mouse colony at Department of Immunology at the University of Washington. And this is where I really learned for 3 years all of what I thought scientists do. So it was a lot of skills in molecular work and genetic work and using cool instruments, but a lot of it also was taking care of this giant thousands of mice mouse colony. So I also learned a lot of sterile techniques. So especially during a time of a pandemic, I'm just automatically trained to see microbes everywhere. So after this is when I really thought, maybe I just don't like science and I'm gonna leave, and I'm gonna go searching for these jobs outdoors now. So I ended up finding a graduate student who was working on looking at how forests and urbanization, and how we have, I don't know if you guys are familiar with like the Cougar Mountain area, Redmond watershed area, and how these wild places have different bird populations in comparison to areas where there's been a lot of development. And this project was really fun because, of course, I'm just trapping birds all day. And we discovered that there is actually more diversity of bird species when you're at the edges of two ecosystems or more ecosystems. So I thought that was really cool to think about this edge effect that's going on. It's not necessarily more species in suburbs or more species in forests. And there's just more niches that they can fill. And so I also started getting involved in tracking where bigger animals were moving along the Cascades. So many of you might be familiar with the I-90 Bridges Wildlife Coalition. So I just saw they have built these bridges now over I-90. This was years ago when I was helping to figure out where animals were trying to cross. And so we would input that data. I think this was considered a citizen science project, because even though we were trained, we didn't have to have science backgrounds to help with this project. So this was one of my first introductions to not necessarily needing to be a professional helping with a scientific project to track. And then I also found another graduate student who needed help with her work. And a lot of these forest ecology jobs I found were very seasonal, so constantly looking for new work every couple months. And this was a project where we were tracking the western gray squirrel, which is very important for distributing seeds and fungal spores around the forests in the Cascades. The squirrel also tends to make nests out of the invasive mistletoe, which will cause, it will infect trees and cause them to sprout branches lower down, which can be a problem when we have the wildfire season. And those wildfires come through and kind of can ignite the forest. So there were prescribed fuel burns to kind of control for mistletoe, but they were wondering if this influences where the squirrels then live. So I basically spent a couple of seasons hiking around Stehekin at the northern end of Lake Chelan in the Cascades, and it was amazing. But you know, these projects always come to an end eventually. So I also turned my eyes towards international opportunities and ended up going down to Mexico for 2 months. And this was just a project looking at how the endangered maroon-fronted parrot, or macaw, tends to breed and use the space on this cliff. So every morning we would hike up to this cliff and sit in front of it and just track what the parrots were doing day in and day out for 2 months and just camp. And it was amazing. But after a time, you realize that you need to make some other way of steady income. So I fell back on my skills that I had years prior in the immunology lab working with the mice. And I set out to find work in a lab. And I thought I was giving up my dream to be in the field and just work in a lab running experiments. But this time I wanted my project focus to be environmental, because I realized I cared about the environment and what's happening to it. I ended up finding a project that was hiring at the Institute for Systems Biology, which is how I met Beck through one of my coworkers there. And I was looking at how increasing levels of carbon dioxide in our atmosphere might affect these phytoplankton. When I got interviewed, I didn't really know much about phytoplankton. I'm thinking maybe you've heard of them through diatomaceous earth. It's this stuff you sprinkle in your garden to kill the insects. But my boss hired me because she liked that I cared about the goals of the project. And so I ended up spending 6.5 years here running experiments. And it was a lot of fun, but I really did miss the field aspect of things. So I would look for ways to spend vacation time actually out in the field. And this is how I got on this journey of going to Africa was one of the places. I realized I could help be a co-leader and bring undergraduate students to, this is Kruger National Park in South Africa. And we basically spent a month living in this Volkswagen van. And we were tracking big cats. So we were looking for lions, leopards, and cheetah. We ended up seeing 101 lions, 33 leopards, and 7 cheetah in only a month's time. And this is some of the first times these students had ever left the US, and we were camping. So it was amazing and really stuck an idea in my mind that someday I would love to run a project like that, where you're interacting with people to learn about the natural world. And this dream to go to Africa again happened when 2 years later, where I went to Zimbabwe. And this time I partnered up with a Zimbabwean researcher who tracks the wild dog, or this painted dog here in the middle. And painted dogs kind of have the same story that our wolves have in Washington, where, or in the North America, where they're seen as a pest. They kill livestock, and farmers don't want them around. So there's a lot of traps that are set to capture them. They just get snared, and there's bush meat traps out. So the researcher was looking at how wild dogs, similar to the squirrels, actually help herd animals throughout these large spaces in the parks. So they're really important kind of control species in that sense. And then, of course, these are all just like vacations that I've done while I'm working on the experiments and phytoplankton in the lab. So these would be like every year I'd go do a trip. Also with Peru, because I love the jungle and forests, I would find ways to get to the Amazon, and we'd look at biodiversity. And this is where I was introduced to this concept of ecotourism. So people will go on vacation as tourists, but they want to participate in science projects. And Earthwatch was actually a big organization, or is a big organization, that was dedicated to that, research-focused vacations. And so it was amazing. You just wake up morning to sunset looking for as many animals as possible on a ship, and then you got to interact with the researcher, which was awesome to hear the lectures in real time. And then I found another example of this in southern Peru through rainforest expeditions, which did a similar partnership with ecotourism. So the researchers are down there looking at the endangered scarlet macaw, blue and yellow macaw, and the conservation efforts while there's deforestation going on. How do you conserve and protect the wildlife and these beautiful macaw species? So you could come down as a volunteer and climb into the canopy and hold these little macaws, and the vet would take measurements, and then you were responsible for putting this little naked looking fat chicken into the tree. So it was a really cool thing to see how you train people who don't have background in this, and then how they just let you go and trust that the data is being collected. There's certain ways they know if it's good data or not. So I kept this ecotourism model in the back of my mind as well. So now we go back to Washington, and I start to try to find ways to get out of the lab and more involved with being out in the field. And maybe you guys have heard of the Puget Sound monitoring program. I started to work on one of the ships that went up and down. It was the Clifford Barnes through the University of Washington, but now it's called the Rachel Carson, is the new ship that they have. And so we could go out and I could get my hands wet, so to speak, learning about the ocean and how to apply what I'm learning in the lab to actually collecting samples from the ocean itself in the Puget Sound, and then monitoring what these phytoplankton are experiencing as, you know, increasing levels of carbon dioxide are coming into the ocean environment. And something happened during this time that totally changed my life. So my boss came to me and said that she was running out of money and she would have to probably let me go. And I was like, oh my gosh, what do I do now with my life? And it was maybe a month later that she's trying to sort something out. And she comes to me and she instead says, oh, actually, I'm going to send you, I just got funding, but you have to go to the windiest, coldest, driest place on Earth, because it's funding for a trip to Antarctica to do research on phytoplankton. And I was just like, oh my God, like I've never been on a real research vessel for that long. It was going to be for 53 days. I always thought I would go as a tourist. It was completely an unpredicted opportunity this is like a make or break moment where I had to go in her place and basically, what do they say? Fake it till you make it. And it was amazing. So I flew to New Zealand, and then from New Zealand set off on a Hercules C-130 down to the Ross Sea, which is, if you were to just go straight from Australia, you would get to the Ross Sea. And I stepped off the plane and they warned us it was going to be cold, but I didn't realize it was going to be so cold that even breathing formed icicles. I didn't have a beard, but my hair would form icicles immediately. And I was just amazed. I thought Antarctica was flat, nothing, just whiteness, but it actually has a huge mountain range that runs through it. There's active volcanoes, and it is amazing. It is a desert environment, so definitely it literally takes your breath away from the moisture, and your skin immediately starts to crack. Your hands are drying out. You need 10 times more chapstick than you thought you should bring. And as soon as we landed on the ice shelf, we are then carted away into this bus, Ivan the Terra bus. If anybody has aspirations of being a bus driver, you can actually apply to be the bus driver down in Antarctica. And being down there, they took us all the way to one of the US research stations. There's 3 of them. There's the South Pole. There's also McMurdo in the Ross Sea. And then there's one on the Antarctic Peninsula, which is Palmer Station. So we were at McMurdo, and there's a lot of history here. It definitely looks like an old miner's town. It's kind of dirty because the snow is melted off of the ground at some points in the year. And you can climb this hill, Observation Hill here, and look out across the expanse of the Ross Sea. We had a lot of time to kill while we were waiting for the ship to come get us. We had 5 days on station. So while you're on station, this is the time to make sure you have any last minute lab things measured out. Because imagine if you're on a ship and you're trying to use a weight scale to measure anything, the ship's rocking. So you have to pre-measure anything you might think you need before you get on the ship. I also learned that there was a marathon going on, so I just happened to have a costume with me, because also these are actually quarantine tips and tricks if you want to take notes, but you just have a costume handy anywhere because you have to keep your sanity and morale high. So a couple of us, there were 25 of us that ended up running a marathon while we were waiting for the ship to come. And I don't know if Northwest Runner, this is an old article, but I wrote an article about the experience. The water bottles just immediately freeze into ice cubes. So that was probably, I would not do that next time. And of course, you're in an extreme environment. So you have to dress warmly. Let me see if this plays. The lowest temperature in Antarctica was actually recorded in 1983 at, I'll do the 128 Fahrenheit or negative 89.2 Celsius. Sorry, negative 89.2 Celsius. And this is in the interior of Antarctica. So the interior gets much colder than the peninsula does. And the peninsula can actually range and go anywhere from, you know, you're talking about winter extremes where it's no lights coming in and it's super dark, to a summer extreme where it's daylight all the time. So the average annual temperatures range from negative 10 Celsius on the coast up to 10 Celsius, 14 Celsius sometimes while I've been down there. Either way, very extreme temperatures. They say Seattle's cold, but it's not that cold. So we spent 53 days at sea. We were looking at all this brown stuff. So this is all of the algae, this golden pancake shaped ice. And we lived on board. There were 25 scientists and then 25 support staff that just work on the ship full time, or during contracts, but full-time contracts. And this is what we're realizing is the forest of Antarctica. So this phytoplankton starts the spring season, and we were down there to look at what happens when it starts growing and then how it sinks to the seafloor and how it interacts with the atmosphere and how different animals might be using this source of carbon or organic carbon from this microscopic forest. And the Ross Sea actually is just a frozen ocean. So a lot of it is, you see the swell, but a lot of it is kind of calm. So if you imagine you're seasick a lot, I wasn't actually seasick most of the time because you're kind of protected by this whole surface skin of ice. This is around 75 degrees south if you're globally orienting yourself. And I wanted to show you a glimpse of the inside of the research vessel. So the United States has 2 US ships. When you always ask how much does it cost to run these ships, everybody always says they're about $60,000 a day. So a lot of Antarctic work requires much more funding than doing science elsewhere in more easily accessible parts of the world. So you, as a scientist on the ship, you can drop this big instrument that you see here in the video down into the ocean, all the way down to 4,000 meters. And all these little bottles that are arranged in a circle can actually capture different sections of the ocean. So you drop it all the way down to the depth that you want. And as it's sinking, it has sensors on it that will tell you different things about the ocean. So how much oxygen there is, how many phytoplankton, not how many, but where you have the most phytoplankton, what the temperature is, how salty it is. And then as it comes back up, you can trigger each bottle to close, and it captures that capsule of water. And then the scientists come in and they fight for who gets to get the water first. And then we bring it into the lab, which we've actually set up prior to getting on the ship. So everybody's responsible for setting up their own station and making sure everything's secure. So you bring bungee cords and you bring little screws to keep everything secure if the ship were to hit into a storm. And I was responsible for looking at these, basically it's like algae. If you think of algae excreting a mucousy goo, it's like their form of pooping, so to speak. That's what I was tasked with trying to measure. And of course, 2 months is a long time to be on a ship. So we did find one day that we were able to get off the ship. We took the ship as far into the ice as we could, and we jumped off and we were able to play soccer. We were able to have a little photo shoot like we were at the beach, and we were able to kind of regain some sense of sanity because, you know, confined spaces, we all experienced this earlier on in the pandemic times. So one of the other really cool things was one of the researchers was interested in penguins. So a large, this group of penguins came to investigate us. I think they're really drawn to the red of the ship and our shirts. You know, they are adapted to looking for krill, this little crustacean, which is a red color. So, and it was just a face-off, and they were curious. They have no land predators. And this was one of the most southern colonies that the professor we were with could put tags on to monitor how deep they dive and where they're going to look for food. So that was amazing experience. But one of the other amazing experiences I had was learning more about the ocean. So I don't know if you guys already know this, but I only learned this in 2013. Was there's a whole conveyor belt around the whole Earth, and it has to do with the equator being hotter and the poles being cold, and the rotation of Earth sets up this conveyor belt, we call it. You have different densities from different saltinesses and temperatures of the water, and it causes this kind of this current. It's where we get our currents from. So because of this, it fuels life in the oceans and on land. It's always replenishing nutrients to the surface, where you can have that microscopic forest growing in the surface ocean. And a lot of the deep, cold, salty water gets produced up here in the northern hemisphere near Greenland, and then also down in Antarctica. So you have, as the surface of the ocean freezes, it forms that skin of ice, and the ice doesn't keep the salt. So that salt is rejected. That's called brine rejection. The salt is rejected back into the water, and that just gets super cold. It's negative, almost -2 Celsius. And that heavy, cold, dense, salty water sinks to the bottom of the ocean's floor. And it's been said that it doesn't come back to the surface for about 1,000 years. So whatever's happening at the surface in the northern and southern hemisphere will be input into the ocean for around 500 to 1,000 years. So it's pretty incredible. And then I always thought the ocean was just a big bathtub of water. And it turns out that my experience with geology, when you're driving and you see geology and you can see all these different layers, my experience with that made sense, that different layers of Earth are different periods in time, or different elements make up different layers. The same thing happens in the ocean. So this is, might look confusing at first, but you have Antarctica here in the middle, and you have the currents circulating around Antarctica, and that generates this mother ocean that then, like I told you, the ice forms and sinks this salty, dense water to the very bottom that travels all along the bottom floor of all the ocean basins. And then it comes back up through that idea of convection, just from the equator and the poles being totally different temperature extremes. And these layers don't really mix vertically, they just slide along each other. So this is how Antarctica is connected to everything that's happening on Earth, and also huge weather patterns that we see. You can actually see these, the mark of this through pictures like this. So this is vegetation. If you were to measure vegetation on land and in the ocean, all plants, as you know, might know, have chlorophyll, or this pigment that allows plants to absorb sunlight and convert that into energy. And if you use a satellite, satellites have sensors that can measure this from space. You can get this picture. This is an image showing the maximum. So in the summer of the northern and the southern hemisphere is pictured here in the oceans and on Earth. And you might think a lot of Earth's oxygen is being produced from trees and plants on land. We have the rainforests. They're amazing oxygen producers, and they regulate the carbon cycle. But it turns out that oceanographers have discovered that phytoplankton living in the ocean actually are responsible for probably more than 50% of Earth's oxygen. And so it's not just the plants on land, it's also everything occurring in the ocean. And you can see where you have the darker green streaks is where you're getting more growth of these phytoplankton. So this is I should probably say, if phytoplankton is a new word to you, it's just two Greek words, phyto, which means plant, and plankton, which means drifter. So they're just drifting in the ocean, and they're single cells. So each one that you see pictured here is its own individual organism. And this is an amazing, mesmerizing video, I think, of the Earth breathing. It's like between the plants on land and the ocean. It's the two lungs of the world. And I could just watch this forever. So you see summer coming in the southern hemisphere, and then the ice coming in the winter, and it oscillates between the north and the south. So I find this fascinating. So many of you might wonder, okay, how is this related to my personal daily life? If any of you have seen bioluminescence, so actually one of the best bioluminescence I saw was up in Friday Harbor in the San Juan Islands. It was magnificent green. There's also the red tides. We actually just had down in San Diego a red tide that lasted more than a month, and it produced this blue bioluminescence. Those are because of phytoplankton. Also biofuels. So a lot of our fossil fuels come from ancient phytoplankton that were preserved in the bottom of the sea floor, and also is being used as a way to potentially have genetically alter algae to make biofuel production. And then also, if you have ever had this kombucha, the multigreen, I actually put some of this under the microscope, and it has cells in it, spirulina and cyanobacteria, or blue-green algae. It says it on the ingredients list, and they are in fact in there. And then it's all because of these microscopic organisms. So when they grow in large numbers, we call that a bloom. So it's like wildflowers blooming in a field, and you have this normal ocean condition. The water looks very blue, and when you get a lot of phytoplankton growing, it turns the ocean kind of a green or a brown or a golden color. And these can actually be tracked from space. That's how I showed you the earlier little video of the Earth breathing. And if you were to take a net that's specially designed for, I should say in that last slide, NASA is really interested in monitoring the health of Earth through remote sensing. So there's a lot of tree projects actually, like monitoring vegetation from space, as well as phytoplankton from space. And if you were to take that phytoplankton, you collect it from the water, you can have this golden soupy color that when you remove all the water, you're left with this pancake of green, and you put that under the microscope and it's these amazing intricate images. And when you look under even higher powered microscopes, you see there's a huge diversity. There's actually hundreds of thousands of species. Some are made of cellulose or wood, some are made of glass, silica, some are made of chalk out of calcium, and they have been around for hundreds of millions of years. So I thought I could just quickly introduce you to a couple of them. If you like naming trees, this will be, just think of this like your Douglas firs or your maples. We have the diatoms, which are the glass houses. These guys are actually responsible for 1/5 of Earth's oxygen. We've got dinoflagellates. Those are whirling whips. They're the crazy looking ones that produce a lot of the red tides and toxins. We have phaeocystis, or floating colonies. These guys produce a compound that actually has been found to inspire clouds to form. So a lot of the reason we have clouds forming is because of algae in the ocean. And if you're wondering what exactly they are, they're not archaea, they're not bacteria, they're not plants really, they're not fungi, and they're not animals. They're this whole group of life called protists, and they're very bizarre. So I just quickly wanted to share this crazy evolution story with you, so to show how old they actually are. So 2.7 billion years ago was when you had your first photosynthetic cyanobacteria. It was engulfed or eaten up by another organism, and that was around 1.5 billion years ago. We started to see red algae and green algae. The green algae is what gave rise to the land plants that we know today. That was 470 million years ago. And then the rest of them got engulfed over and over and over again until we got this crazy diversity. And now you have like Russian dolls stacked in with each other. If you were to look at the genetics of these organisms, one of them has a genome 100 times larger than the human. And you're wondering, what is it doing with all of that information packed inside this cell? So if you're a sci-fi fan, this is what really gets me. There's so much more to always be learning about these guys. But I definitely sat back in the lab and I was thinking, man, how do I learn about these things, but also get back to Antarctica? Because Antarctica is what I really can't stop thinking about. And I decided that it was time in my life to go back to graduate school. So I ended up moving from Seattle down to San Diego to do a master's degree in marine biodiversity and conservation. And before I get to the next segment of the story, which will be short, I just wanted to see if there were any questions from the audience that I should field.
So at the moment, not a ton. There's a couple questions that we're going to have about the citizen science and the effects of melting glaciers and whatnot on the phytoplankton. I think you may get to those things. So I think we'll hold on to those until the very end of the question. Actually, there is a question that just came in. So first off, please tell Allison that she is so wonderful and inspiring. And in order to hear her awesome Talking Antarctic Gold, someone from Russia had to wake up at 2:30 a.m. to join the webinar. Here is the question. How many species of phytoplankton grow in the Southern Ocean, and what are your 3 favorite plankton species?
Oh my gosh. Okay. Thank you. I think that's Anya. I am impressed you woke up for this. Thank you. There are, I think 140 some genera that are in Antarctica. I don't know how many species because they're still very difficult to determine what is a species. My 3 favorite would probably be the diatom, which uses the glass to make really intricate shells. And then the dinoflagellates, because they're the ones that will cast a mucus net around whatever they want to eat and digest it and slurp it back into their cell body. And I think that's really bizarre. I'll just go with those two for now as favorites. So.
On. Perfect. Great. Thank you so much for that question and for waking up so early, Anya. We really appreciate that. And I think with that, we'll go on and finish on through and then close out with some more questions for you.
Yeah. This ends at 6, correct?
We can go a little past. There's not a hard stop.
Okay. So let's jump back down to Antarctica. I'll actually show you where I was on the continent. There are 2 ways to get there. You can take a ship, which is about 2 days crossing over the Drake Passage, or you can fly, which is about 2 hours, 2.5 hour flight. And that's from South America down to the peninsula here. So down in Antarctica, the ecosystem looks like this in cartoon form. It's very simple in comparison to other ecosystems. So you have the ice, and as the ice melts, you have sea ice and you have glacial ice. It fuels this phytoplankton, which I've been talking about. The phytoplankton is then eaten by krill, and then that's eaten by the whales, seals, and the penguins. So there's actually a seasonality to this. So just like you have plants on land come at different times in the season, you also have different phytoplankton that come at different times in the season. And one of the things we're interested in looking at down here is the fact that the Antarctic Peninsula is one of the fastest warming regions in the world. So of all these glaciers, researchers found in 2005 and again in 2016 that 87% of these glaciers are retreating. That means they're not accumulating as much snow as they are losing it. So in all, their balance is off and they're basically moving away from the ocean environment. So down in Antarctica, I also learned that there is a growing tourism industry. And I thought, okay, if we want to address this issue, maybe we could involve the tour community, because scientists can't get down there the whole 5 months of the daylight season. And you have many ships that are down there and it's growing. So back in 1995, you maybe had 6,000 people that were coming down to visit. But then just this last year, there were 57,000 plus that actually stepped foot on the peninsula. And so I just thought this is a really cool opportunity. And that would be through the platform of citizen science, which is just non-professional scientists getting involved in a scientifically driven project. And so if you want to compare one research vessel, which is $60,000 a day, to a program you could buy simple oceanographic equipment and have people help you collect phytoplankton from the surface of the ocean, that's only maybe $20,000 to put that equipment together and to do the analysis, but you're covering 5 months of time and you can reuse that stuff over and over again. So, and you also get to interact a lot with the people on board, which is amazing. So I wanted to play, do we have time for this short 8-minute video, or should I say?
Let's we can post that for folks later. It's a great video and I encourage folks to watch it. But just in the interest of time, let's move forward, and maybe after questions, if people want to stick around, we can play it for them then.
Okay, sounds good. So basically, I work with the tour guides on board the ships to train them how to take samples in the way that we want them collected. And then they are the ones that will engage all these travelers in the project. So everybody gets a little task to do, and they have to help, or they get to help. They can help. They're not forced to help collect phytoplankton. And I think it's one of those, it ties in everything I had been working up towards in my personal career of being in the field, engaging with people, getting everybody involved and excited about science, and now working in this phytoplankton aspect to it to try to make these unseen heroes of the Antarctic ecosystem come to life and show people that Antarctica actually is rich in life because of these phytoplankton. So one of the results we have from the project so far is this a very colorful graph. So if you were to imagine each group of phytoplankton gets its own color, and then you can just look over time from the beginning of the season, November to March. So this is the summer down in the southern hemisphere. And you count how many of each kind you see along the peninsula. You notice just quickly glancing that there's a lot of blue. So you would think, oh, maybe this group, the flagellates, are important. But when you convert this number, you can also use the shape and size of a cell to determine how much carbon or energy it would be providing to the environment. You actually see that it's a lot of gold. The gold color are the diatoms, those groups I told you about that use silica glass to make their frustral. So the ecosystem is heavily relying on a lot of these diatoms for energy. So the krill, this little crustacean that eats phytoplankton, really selects diatoms. And we can also notice that sometimes in the year you get spikes in red group, the yellow group, the green group. And if we pick these out, we're able to look under the microscope to see who they are. And we discover that it's actually a couple different types of phytoplankton that really haven't been documented down there. So because of citizen science, because these samples are collected more frequently throughout the whole season, we can see these things that aren't necessarily written about a lot in the literature, in the scientific literature. So far we have samples collected over 4 years from all of these locations along the peninsula. And when those samples come back to us, we look under the microscope. There's a student in Argentina who's helping with this project. And then I take these, I was able in June, with COVID testing on a weekly basis, I'm able to go into the lab and I could process the samples that were collected on a filter and look at a new tool that we have in oceanography is to use genetics to identify species that are down there. Instead of just looking under a microscope, you might miss something. We can use a DNA sequencing approach. So this is just showing me converting the actual sample from the ocean into hundreds of tubes. I had 400 tubes, and then each little white band shows me that it worked, that the genetics part worked, and I'm getting the data back now. So the next chapter of my life will be sitting on the computer analyzing that. We've been able to publish so far in 3 different journals. So I put the QR codes there if anybody wants to take a picture of those. You can hold your phone up to them and it will link you to the links of each article. But this has been exciting because the idea is to share that this is an amazing collaboration that's going on down in Antarctica. And so what I'm thinking of when I'm trying to put this whole story together is the forest. So all my experiences and what I know about forest ecology and on land, how is that now going to relate in a 3D environment in the ocean with thousands of species we don't really know a lot about, and as a source to fuel this ecosystem. So, and then also how is the melting glaciers from land going to influence what types of phytoplankton we see? Because just like trees, each phytoplankton cell actually has a preferred environment that it likes to live in. So I hope that the next time you look out at the ocean, you think of the invisible forest and you give a hoot about the little guys, the phytoplankton. And with that, I just want to say thank you to Friends of the Forest and for having me speak and for your attention. And also, of course, everybody who's been involved in this project. Scientists do not work alone. We work in teams. And so this has not been my spearheaded efforts. It's with a big group and some obviously donations and funding sources from the government. So thank you. I can take questions if anybody's interested in following the project or me on social media. I'm on all these platforms, and then we do have a website where I try to put blogs up. So if you want to stay tuned with what happens over the next 4 years of my PhD.
Right on. Thank you so much, Allison. That was wonderful. And we do have a couple questions if you got some more time for us here. Yeah. So Anya also asks, and you kind of started touching on this towards the end, but how the continent warming will affect the life and quantity of phytoplankton.
Good question, Anya. So So phytoplankton, we always say there's winners and losers because you have so many different species. Some might do better than others. So the diatoms, for instance, a lot of the experiments I was doing in the lab years ago were showing that they prefer, it's like carbon dioxide is fertilizer, and warmer temperatures, they love that. So they might do well. But other areas along Antarctica, they might not have that same effect, because you have to remember there's so many species kind of acting in concert together. So I think, yeah, this side of oceanography is still relatively a new frontier. So it's conflicting evidence for who exactly is going to win out or do better. But the idea is that smaller phytoplankton prefer fresher water in Antarctica. So we might lose these big diatoms that were the Antarctic gold that really fuel the ecosystem.
Awesome. We do have a request. If you could put that last slide up so folks could take some pictures and make sure they get that info, that would be great.
Yes. Whoop. This one. That one, right?
I believe it's that one. If it's a different one, let us know, and we can put that up too, folks out in the audience there. So question from Robert: why should we in the Northern Hemisphere care about a single-celled creature in Antarctica?
Good question, Robert. I would think, well, a good answer might be because the same phytoplankton that are living down in Antarctica are also living in the Arctic. So you might have a different ecosystem, but the first layer of life is very similar. So what we learn about down in Antarctica can help us understand what we learn about in the Arctic for polar bears and cod and seals and walrus up there that are feeding on copepods, which are feeding on phytoplankton. So I think anybody who's interested in healthy marine ecosystems would be interested in however much we learn about phytoplankton anywhere in the world.
Great, thank you. Bill wants to know, do higher organisms like krill get nourishment from these diatoms made of glass or silicon? Seems not nourishing.
You don't like eating glass shards? Yeah, the diatoms, especially the ones down in polar regions or up in polar regions, are really good at being fatty, rich in lipids and fats, as a way to stay afloat and as a way to have nutrients available to survive the winters. So these krill or crustaceans really do target the diatoms for those nutrients and fats. And they have processes with dealing with the silica, so it doesn't really affect them. I would imagine it's similar to like birds having a, oh my gosh, I should know the word. I'm blanking on the word that helps them grind. They have the grindstones to help them deal with if they're eating clay, for instance. It's a similar process in krill. So
awesome. And actually, the slide that folks want to see is the one with the journal articles.
Oh yeah. Okay. Zoop. That one.
That's the one. Great. Thank you. We have someone who asks, why did the macaws that you told us about have no feathers?
They are born that way. They're born naked and they're tucked away in a little nest. So their parents lay on them to keep them warm. They don't need their feathers yet. Their feet are the first thing to grow. So they have these giant feet, and their beaks are giant because they have to eat the food their parents bring back for them. And then eventually the feathers come in. It's the last thing to come in as they're growing, maybe a month and a half, 2 months, 3 months in, and then they start to look really raggedy, and then they can start to try to fly away.
So they do eventually get their feathers.
Yeah.
There's a children's storybook there. I'm not quite getting through it, but it's there. All right, have you found any cousins of, quote, cousins of phytoplankton that live up in the Pacific Northwest down in Antarctica?
Oh, cousins. I mean, a lot of the same taxa or groups, those big groups I was telling you about, like in the Puget Sound in the Northern Hemisphere, you have those same diatoms, dinoflagellates. The major groups are found in a lot of areas. It just might differ in which species you find. But yeah, the Puget Sound's full of diatoms and dinoflagellates and Yeah, Pseudonetia is one of the ones responsible for the shellfish farmers having to close down if there's ever any toxic blooms happening, which I think it usually happens like late summer, fall.
Right, cool. So this is a question from me. So the citizen science side of things, are there other areas of science that have seen such success with it that you're aware of? Is it being used widely, or is it kind of a new, I know of other areas it's used, but to the same effect?
Yeah, there's actually, so the term is relatively new, but like maybe within the last 5-6 years, to really like the Citizen Science Association exists. There's actually scistarter.org has over 3,000 active citizen science projects around the world. And so but this has been a thing that's been since the beginning of actually weather prediction. It was just people in their backyards having rain gauges, detecting how much rain was coming, and it helped inform the weather forecasts that we have today. So that was a program called COCO-RAS. It's a big acronym. But there's also Cloud in the Crowd is an awesome free TV series about many successful citizen science projects and big ones that have helped. The Flint water crisis is considered a citizen science project where the citizens themselves or the local residents helped determine that the water was really bad. So, yes.
Very cool. We actually have a citizen science project going on in the forest lands right now with Transition Fidalgo is leading it and doing some monitoring in the forest lands. So if you haven't checked that out, make sure you go to Transition Fidalgo's website and check out all the cool things they're doing. Allison, this has been awesome. I just can't thank you enough for taking the time to talk to us and telling us about your adventures and your story and all this cool stuff about Antarctica. For folks that are watching, we are going to have this posted on our YouTube channel, so you can come back and check it out and watch it again if there's some things that you missed. I know that Beck follows Allison on Instagram and is one of Beck's favorite people to follow. So make sure you check out Allison's Instagram if you're on that as well. And if you don't mind, Allison, if folks are wanting to stick around a little longer, we can play that video. And if you need to go, thank you so much for joining us. Our first night of our Give a Hoot fundraiser, our fall fundraiser this year. Like I said, we've got a bunch more coming up, so make sure you check out the website friends-of-the-forest.cosvox.com. And Allison, do you think we could watch that video?
Yeah, we can for sure. Thank you again to everybody who came tonight, and good luck with your event this week.
Thank you so much. Thanks, everybody. We'll see you soon.
I'll cue up the video now. Antarctica and the polar regions in general are just very remote parts of the world that are very difficult to access, so it's just very data limited regions of the world. There's a lot we still need to know about them. They're also the fastest changing parts of the world, so there's an importance to understand these ecosystems, but how to do that is also very challenging. Fjord Fido is a citizen science project that takes place down in Antarctica in the polar regions. And it's in partnership with researchers, so with the Vernay Lab and myself, and a student in Argentina, partnering with tour vessels down in Antarctica.
Citizen science can mean a lot of different things, but fundamentally it is a research project that involves science, and people that don't necessarily have a scientific background are helping to collect an increased number of data points.
Before, there was no citizen science per se. For me, it only started 4 years ago. We started teaming up with the tourist ships and teaching them how to collect samples, so that then we will have now samples collected in times that we are not there. And the project that Allison has, for example, on phytoplankton in the Antarctic Peninsula, would not have happened without the citizen science.
Phytoplankton are, I think, amazing little creatures that we don't even really know much about in the sense of your daily life. So they live in the ocean. They are actually contributing to global processes. So in one drop of seawater, you might not think there's much going on, but when you look under the microscope, there's all of these organisms, and together they're contributing over 50% of Earth's oxygen, so that's more than the plants and trees on land combined. They're regulating carbon that moves from the atmosphere down into the ocean's depths. And so I think when you start to realize there's these little single-celled underdogs making these huge things happen in Earth as a whole and giving life to life is amazing. So if you break up the word, it's phyto and plankton. They're both derived from Greek. So phyto is just plant, and plankton is a drifter. So they're like little tiny plant-like organisms drifting in the oceans. They're also just incredibly intriguing to me because they're ancient. They've been around for hundreds of millions of years.
The phytoplankton is the most important thing in the world. The same as humans depend on crops, the phytoplankton is also the food for all the animals that live in the ocean. So you have the phytoplankton that is only one cell is being eaten by another little animal, and that little animal is eaten by an animal that is a little bit bigger until we'll get all the way to seals and penguins and whales. So we call that the food web, the marine food web, and the phytoplankton is the one that makes it possible. Without it, none of these beautiful animals that we love would live.
So we get a group of excited people to come in the boat with us. We call it the science boat. And while we're out there, each little task gets delegated to a different participant. So somebody, of course, has to record the data. That's very important, to be the scribe. Another person will drop a Secchi disc overboard to get a quick visual of when this disc disappears. That's kind of when the light levels are low, and we call the Secchi depth. Another person can drop an instrument overboard that measures the saltiness and the temperature of the water from the surface all the way down to 60 meters depth. And that tells us what might be changing in the marine environment, and if phytoplankton have a specific preference for where they're living, then that's important information. And then we'll take, just dip a bottle overboard so we can get a volume of water and be able to count how many phytoplankton we see in there. And then we also take the net and we tow it behind the boat to concentrate the phytoplankton, and then this gets preserved onto a filter so we can have enough material to extract the genetic information from the organisms. We always say that scientists need to get out of the ivory tower and interact more with the public. If science starts with observation, then people can help with science through observations, and you just have more eyes on the ground, collecting more information about how the world works. For Antarctica specifically, I was really struck by the fact that in the summer you have maybe 5,000 researchers from all the nations working in Antarctica, but then on the peninsula you have this tourism industry that's growing with over 59,000 visitors coming, so that's more than 10-to-1 ratio of visitors to scientists. So for me personally, that was my motivation to get that community of people involved in the polar research that's going on down there, because Antarctica is preserved as a place for peace and science.
Antarctica is the only continent in the planet that has an Antarctic treaty. It doesn't have a government. It doesn't have a passport. It doesn't have a nationality. Everyone that is going to respect the place is entitled to go there. So in a certain way, this is utopia. This is what we all aspire to, that many countries come together, they agree on how to interact. So that international sense of community, it has that sense that it can go beyond our human limitations and create something better for humanity.
The best part about participating in citizen science is having the opportunity to share this rare facet of science with someone that is seeing the project almost with childlike eyes, things for the first time, and it puts everything into a different perspective. And as a scientist, it makes you question things differently because you're being confronted with questions or thoughts that are more often than not taken for granted. A project like Fjord Fido brings this more profound experience to the forefront of how the world is dependent on these organisms that aren't typically studied or given a lot of spotlight time. I love getting more people involved with citizen science because I like to think that it starts this spark of curiosity in their world that otherwise wouldn't be there. And the more sparks that you have, eventually it will add up into potentially a roaring fire that can really drive actionable change. Because at the end of the day, people care about what they know, and if they don't know about it, then they probably won't care.
Right on. Thank you so much, Allison. That was great. Thanks again for being here with us. We look forward to hopefully having you back. Maybe in a future time you can do an in-person talk when we can all be in one place again.
Yeah, I would love to see you guys in person and get out into some forests up in the Pacific Northwest.
Yeah. How many years you got left in San Diego there?
Hopefully 4.
Uh-huh.
Or
All
less.
right. Well, 2024 then.
Yeah, I'll be there.
All right. Right on. Thanks again. And we'll be sure to post all this stuff on the website so folks can follow you and support your work. And hopefully we'll talk to you soon.
Thank you. Hope you guys have a great week.
Yeah, you too. Thanks, everybody.
Bye.
Bye-bye.